Hello guys, welcome to our seminar about the article A New Circulating MicroRNA for the Detection of Acute Myocarditis. My name is Sara Osorio and she is Natalia Ojeda. We are medical students and today and this time we will tell you about an interesting study, so let's get started. What is acute myocarditis? Myocarditis is an inflammation of the myocardium, the cardiac muscle. Acute myocarditis is most commonly caused by a viral illness. However, it may less commonly be caused by non-infection etiologies.
What's microRNA? MyRNAs are a class of non-coding RNAs that play important roles in regulating a gene expression. The majority of mRNAs are transcribed from DNA sequences into primary mRNAs and then processed into precursor mRNAs and then finally mature mRNAs. In short, they regulate gene expression post-transcriptionally. With this article, we sought to identify the novel microRNA for the diagnosis of acute myocarditis.
In this article, they used a lot of methods. One of them is PCR. It is a molecular technique that is based on the enzymatic amplification of the genetic material and that is based on the properties of DNA polymerases. To replicate DNA strands, using high and low temperatures to separate the new DNA strands after each replication phase and then let the DNA strand come back together so they can be duplicated again. In this article, it was used to identify the presence of microRNAs under the study that were associated with acute myocarditis. With this test, it was detected microRNAs that were synthesized by Th17 cells and specific microRNAs of plasma of mice and markers in patients.
Then we have the knockout. The knockout is an organism that is generally being genetically modified to specifically turn off one or more genes. Scientists create knockouts, usually in mice, in order to study the impact of a gene loss and to learn about its function. It's a genetic technique that consists of suppressing the expression of a specific gene in an organism, and so we obtain that it doesn't express the gene in a specific tissue or the whole organism. This study was carried out in mice with a knockout in the gene that encodes the CD69 antigen in regulatory T cells to inhibit the regulatory action carried out by the transmembrane protein in order to achieve an exacerbated immune response to the mice using this study with myocarditis. Then we have the murine model that uses the specific and special strains of mice to study a human disease or a condition and how to prevent and treat it. It's done in this study so it can aid into the development of a more specific biomarker for the diagnosis of acute myocarditis and make sure it's safe to then become useful on humans.
Clinical test. The objective of this article was to detect a microRNA for myocarditis. For this, the author carried out a cohort study that included various population or types of patients who initially suspected myocarditis. With this preclinical test, the objective was identify patients that has disease and which other conditions such as myocardial infarction, acute myocardial infarction with non-obstructive coronary arteries, and autoimmune disease. This was done to classify patients and to compare populations. For example, cardiovascular magnetic resonance. It was done to give a final diagnosis of myocarditis. Electrocardiogram. It is a visual representation of electrical activity of hair as a function of time, and in this case, it was used as a diagnostic test for acute myocardial infarction. Endomyocardial biopsy. It is a technique to diagnose myocarditis that is done by removing a sample part of a heart muscle to evaluate the tissue inflammation. Echocardiography. It is a non-invasive diagnostic imaging technique that helps to know the size and the shape of heart. Results. The results obtained after performing all these methods were represented in some graphs that, like the ones we see on the screen. For example, panel A shows us a real-time PCR that amplifies a sample genetic material. In this study, PCR was used to measure the expression of transfected genetic material in healthy patients, in patients with myocarditis, and in patients with acute myocardial infarction. We also have a positive control and a molecular weight marker. And let's see this, that is important, at 40 base pair line, patients with myocarditis have a similar band present to the control. The control band is one that is transfected with microRNAs, and that means that patients with this condition have a presence of microRNA opposed to the healthy patients or patients with infarction. And at the right side, we see a histogram that shows that patients with myocarditis have a greater presence of microRNAs than patients of the controls or that have an infarct. Panel B shows the cloning efficiency because in the PCR, what was done was the clone or multiply the genetic material many times to take advantage of the sample. This tells us how much of this genetic material was cloned in healthy patients and in patients with myocarditis. Therefore, this figure shows us how many colonies were found, having as a result that it was easier to obtain the genetic materials in patients with myocarditis than in plasma of healthy patients because these microRNAs are expressed more in myocarditis.
Panel C then shows the validation of the human homologue of the mRNA found in mice. So, at the top, we can see a lentiviral vector that expresses within the sequence of the human homologue of the mRNA and its location, which is the 8th chromosome, thus the name. Then, a coimmune precipitation of the RNA fraction that was performed with Argonaute 2, which is a protein that contributes to the mRNA maturation and maintain the chromosome integrity, and the IgGs of class 1, which is an antibody that brings protection to the body for viral or bacterial infections. The qPCR analysis then shows the Argonaute 2 is majorly expressed on the fraction with the homologue mRNA, which we can confirm down here. The relative expression of the Argonaute 2 was really high. This brings us to the conclusion that the mRNA was processed and mature. Then with panel C, it shows us the functional validation of the human homologue of the mRNA. In here, what was done is that the reverse complementary sequence of the mRNA was cloned in a luciferase dual reporter vector, which is used to prove the effect of the RNA modification on RNA traduction. We can see that in supernatant of the peripheral blood leukocytes obtained from patients with myocarditis and the expression of the mRNA, the luciferase activity was inhibited. This supports the conclusion of the panel C, which was that the human homologue of the mRNA was a mature and functionally active mRNA.
At the discussion, we have authors and statements. All these comments from authors that were referenced and compared in this article coincide with the team development because these include microRNAs and myocarditis, which are the two central aspects of the article. It mentions the importance of microRNAs as a biomarkers in heart disease, and in addition, the last statements give relevance to the conclusion and results obtained because the study satisfies a lack diagnosis in clinical practice.
Now moving on to the conclusions, then we can verify the importance of the microRNA when providing a differential diagnosis between patients with myocarditis and healthy patients, or patients with acute myocardial infarction or other cardiovascular pathology. Then it makes clear that human genetics is a field in which there's still much to be explored, but that progress is being made every day to find answers to problems by searching and studying the genetic material and information within an organism. And then the importance of the application of the molecular biology concept in medical practice is emphasized thus being able to understand the fundamentals as well as the reason of the use of these techniques to provide us with a better understanding of them and make sure of the use at work from doctors to patients. This was the end of our seminary. We hope everything was clear and that you enjoyed it. Thank you very much.